Welcome, ladies and gentlemen. Today we're going to start talking about macromolecules, so sort of incorporating all of this idea of organic chemistry into what that really means from a biological standpoint and really diving into the biochemistry now, where we're talking about these big macromolecules, these big important compounds in living things and what they look like, but also what is their driving function in living organisms. So let's first just kind of zoom out. And if we're going to talk macromolecules, what does that mean? Well, hopefully you know that that prefix macro means large. So we're talking about these really big molecules that really are formed by smaller subunits. Um, and we'll talk about how these get built and broken down in your body, but they generally fall into four major classes. Your carbohydrates, your proteins, and your nucleic acids are truly, by definition, macromolecules in the sense that they are built from smaller pieces, smaller components. And then we also throw your lipids in here because they are also largely major in their function in living organisms as well, but they don't have the traditional structure of building pieces like the other three classes do. So what we're really talking about here is that these macromolecules are polymers. They're large molecules consisting of many smaller pieces, and those smaller pieces are those monomers, those building blocks. And those monomers may be identical as we link them together, but oftentimes they are just similar to each other. So it's sort of like thinking of these as Legos building a Lego tower, right? So each individual Lego may be slightly bigger, slightly smaller, slightly longer, different colors, but they all fit and piece together to make a larger structure. And that's the idea here. So those monomers most of the time aren't truly biologically active in and of themselves, although there are some exceptions to that, especially when within the carbohydrates. Most of them need to be linked together into that polymer to really truly make a functional molecule, with a few exceptions. So we talk about this idea of stringing those monomers together via polymerization to make that polymer. And what we're really doing here when we're building and breaking down these polymers is going through chemical reactions. So when we build and break down polymers, it's really all going to connect back to that water, right? H2O. So all of the monomers of these macromolecule polymers contain hydroxyl groups and hydrogens. And so by pulling off an OH from one end of a monomer and a hydrogen from the other, we end up with water either coming in or going out, and we thus can utilize the empty bonds by removing those hydroxyls and hydrogens and link them together, or we can add that water and split it to split the bond, remove the monomer from there. So hopefully you know the difference between dehydration synthesis and hydrolysis. So dehydration synthesis is exactly what it sounds like if you break that term down. If you're dehydrated, you are at a lack of water. You've removed water from your body. So when we talk about dehydration synthesis, that's exactly what's happening. We're utilizing the removal of water, which we're seeing right here. So an OH group comes off one monomer, hydrogen comes off another, water goes off somewhere else. And we now have a blank bond on either of these ends of the chain, so we can link them together and create a longer polymer. So we're literally dehydrating those monomers in order to create a bigger polymer. Hydrolysis is the opposite, and if you know what this word means, hydro is water and lys means to break. So I'm adding water to break bonds now instead. And so we'll split that hydrogen into an OH, which will go on one direction, an H on the other, and now I've split this bond and off go the pieces. Heads up, you may come across dehydration reactions as dehydration synthesis reactions, dehydration reactions, condensation reactions. Just know that all three of those terms are interchangeable, and they all refer to the creation of polymers. Hydrolysis tends to be pretty consistently the only term you come across for the breaking of them. So one more time, Dehydration, synthesis, dehydration, condensation. They all mean to build a polymer, and hydrolysis is the one that breaks them. So when we look at polymers, there's a huge diversity in them. Just like we've talked back in organic chemistry in chapter 4, the fact that there's this immense diversity in carbon molecules, well, we utilize that diversity in carbon-based chemistry to really be the underpinnings here for our biochemistry as well. So each cell can have thousands of different macromolecules. Those macromolecules can vary within a species, between species, even within the same cells of an organism, depending on the function of that cell. So the fact that we can build these really large, diverse sets of polymers from just a small subset of monomers really is a driving piece behind the evolution of all of the diversity that we see on Earth. And we're going to take it all the way back down to the DNA level eventually when we start talking about how do I put these nucleotides together, these A's, T's, C's, and G's to make that DNA code, which then translates into all of these unique proteins, which then translates into all of the diversity that we see in living organisms here on Earth.